Hi, and thanks for watching this video on an evidence update for virology, immunology, and therapeutics in relating to COVID-19. Brief outline of this presentation, I'm going to talk a little bit about the biology of the coronavirus, its relationship to ACE receptors and cardiovascular disease, what we know about the immune response, some detail on the potential therapeutic agents that we've got, a very brief look at some aspects of supportive management, and then the evidence for vaccination development. So I'm going to start with a little bit of biology. So this is from one of the first papers published in Nature to detail the genetics of the coronavirus. And they found that it was 96% related to bat coronavirus and was likely transmitted via an intermediate host. That picture in the right-hand corner just shows the horseshoe bat from which it likely came. But the intermediate host is currently unknown. This is a nice image of just what the virus structure looks like, showing the external spike proteins that are important for cell entry, as well as the other surface proteins and the RNA there in the center of the cell. So the diagram on the left here shows the similarities and differences between the current coronavirus and the 2002 SARS coronavirus. But there's great similarities in terms of their attachment receptor and the use of spike protein in infecting cells. And both specifically use the spike protein to bind to the ACE2 receptor. And the diagram on the right just shows this attachment effect and activation using TMPRSS2 surface protein, which enables it to enter the cell overall. And this mechanism is quite well studied already. This is just a couple of nice, more detailed diagrams. On the left shows the diagram from a publication in Nature, on the bottom showing in green the structure of the ACE2 receptor. And the red areas highlighted there are the areas which the coronavirus binds to on the ACE2 receptor surface. And the diagram on the right just shows this again slightly more zoomed out, with the surface protein extending from the virus and interacting with the ACE2 on the human surface cell. It's just nice to see this kind of level of detail. I'm going to touch very briefly on ACE inhibitors, ARBs, and COVID-19 because it's been such a big topic of discussion. If you're interested, I'd really highly recommend looking at this paper here, which was published in the New England Journal recently. The diagram on the left shows how the coronavirus uses the ACE2 receptor to enter into a cell. And the right part of the diagram shows the relationship between the ACE2 receptor and other aspects of the angiotensin system. Overall, the physiology of this is quite complicated, but ACE2 it counters RAAS activation. So it's the counter-regulatory receptor, which degrades angiotensin 2 into angiotensin. So overall, it has the effect of reducing vasoconstriction and sodium retention. And this ACE2 receptor is expressed all over the place, particularly in the lungs, but also in the heart and the kidneys, and in some of the bowel. It's important to note that ACE inhibitors aren't really related to these ACE2 receptors. There's mixed information about the interaction with ARBs on these receptors. So there's some evidence from animal models that they may increase ACE2 in tissues, but this is very uncertain. And in humans, they've only measured plasma and urine concentration of ACE2 activity, and there's mixed evidence on this anyway. So the interaction between ARBs and ACE2 receptors is unknown. And overall, the pathways between all of these things is very, very complicated. The conclusion from the authors of this paper is that they weren't very convinced that the increased mortality related to high blood pressure and cardiovascular disease was not primarily confounded by the age of the people, rather than the use of these receptors and pathways. So more work needs to be done to clarify that. And their overall conclusion is that the data are too limited to support or refute any suggestions to either withhold or prescribe ARBs or ACE inhibitors in COVID-19. However, they recommend that if a patient is already taking ARBs, these should be continued to prevent a kind of withdrawal worsening of their hypertensive or heart failure symptoms. So there's surprisingly little evidence about viral dynamics in the immune response overall, partly because making a test to measure immunoglobulins is quite difficult and time consuming, particularly testing whether it works or not, as we've seen the palaver with the British government trying to get an accurate test. But I'll just run through some of these, which are quite interesting. So this was one of the first papers looking at the immune response, very small number of patients, 34. The graph on the left just shows IgM in blue and IgG in red. IgM was present by 2 weeks in all patients and peaked at 3 weeks and then slowly reduced. And then IgG rises between weeks 1 and 3 and peaks at week 5. This result has since been largely confirmed in this slightly larger study of 173 patients. They found seroconversion with total antibody in the majority of patients, slightly less in IgM and about 2/3 with IgG. Seroconversion times were on average by day 11 for total antibody, IgM day 12 and IgG day 14. At 1 week, less than 40% of patients had antibodies, which increased to 100% after day 15. So this is why the course of the disease is 1 to 2 weeks long, because antibody response takes that long to kick into action. And the figure on the left there, figure 1A, just shows the cumulative incidence of IgG in the samples and showing how this gradually increases over time. This study was recently published and just shows a slightly longer period of follow-up. 60 patients, follow-up time of 6 to 8 weeks. 100% of their study had IgG, and that this was correlated with clinical improvement both from radiology and symptoms. However, interestingly, they note that the IgG titer decreased over time, and the implications of this are unknown but needs following up. And this is another one that just looks at the correlation between immunoglobulins and viral RNA. So again, in a very small number of patients, in 43 patients. So if you look at the top, the black triangles, that's the IgG response, which increased over time. The IgM response, which again peaks by just after 2 to 3 weeks and then slowly decreases, and that the viral RNA decreases as both of the immunoglobulins decrease, as you would expect. This is a very, very detailed case study of the first 5 patients treated in France. And it shows the viral swab results from nasopharyngeal swabs and the kind of viral clearance from nasopharyngeal secretions. The diagram on the left shows the clinical course and events as part of a time series for each of those patients. And it's interesting to note that patient 3 in the middle there is the only patient that died. The chart on the right shows the change in viral load from these nasopharyngeal swabs, which decreased in all patients pretty quickly between days 10 and 14, which is consistent with the kind of immunoglobulin response that we've seen before, except in patient 3 who still had viral shedding after 3 weeks. Before they ended up dying. And although it's a very small study and it's difficult to generalize, there are some similar studies published in Chinese populations as well. So it's worth looking at the viral dynamics and immune response as further papers are published. All right, I'm going to do a blast through therapeutics that are available for COVID-19. Almost all of this is based on this really excellent and thorough review by James Sanders and his colleagues published in JAMA. I'm largely going to use this as a basis and then compare as we go along interesting aspects that aren't included in this paper or new evidence that has emerged since this has been published. So this is a really nice diagram showing the viral replication cycle within a human cell and also the mechanisms of action of potential therapies. So I'm going to start with chloroquine, about which everyone has heard quite a lot. Chloroquine has multiple mechanisms of action and has previously been discussed in relation to the 2003 SARS outbreak as well. So it interferes with the ACE2 receptor. It impairs endosomal acidification and absorption of the viral particles into the cell. And it also has immunosuppressive activities, particularly against TNF-alpha and interleukin-6, which would be important in the immune dysregulation response that we've seen in some of these patients. So what is the evidence for the use of this? In the Sanders review, Mainly the clinical benefits were from media reports or small non-randomized trials. Very limited benefit, especially in severe disease, and any benefits need to be established with randomized clinical trials. This is the second paper there is a systematic review by Cortegiani in the Journal of Critical Care. They note that there's 23 registered clinical trials in China, and also the big European recovery trial has just started as well, which includes an arm looking at chloroquine. They again just summarize the very limited evidence that was available at the time that lots of these discussions around the benefits of chloroquine were being discussed in the press. And then this bottom study here I've just included to show these kind of studies. This was the study published by the team in Marseille, and is a very small cohort of 80 patients. They published it very early. It wasn't randomized, it wasn't blinded. There was differences in the way they treated the patients in the different groups, with azithromycin added in some cases and not in others. They had a surrogate endpoint, which was viral clearance, and they didn't really measure any clinical outcomes. So these are the kind of studies that we're getting about chloroquine at the moment. And we need longer term follow-up and randomized clinical trials to show any specific benefit. So I'm going to talk briefly about antiretrovirals, which are either kind of protease inhibitors or polymerase inhibitors. In the Sanders review, they talk about quite a few of these. I'm going to focus on lopinavir and ritonavir, which is in a combined tablet called Kaletra, which has been used to treat HIV for a long time. There was some evidence of clinical improvement when used for SARS in the 2002-2003 outbreak, but there's limited case reports. I've just included at the bottom here the study by Cow and colleagues published in the New England Journal. So this was a randomized controlled trial of Kaletra in 199 hospitalized patients, so still relatively small. Inclusion, they had to be quite sick with significant hypoxia. However, they found no difference in time to clinical improvement, with hazard ratio of 1.24 and a wide confidence interval, and no difference in mortality. So these are the kind of case reports that we're getting for Kaletra. Ribavirin has also been widely discussed. It was also used in the SARS outbreak with inconclusive results, and there's no results of ribavirin in COVID-19, partly because for its antiviral activity, you have to have very high doses, and this causes significant risk of liver toxicity, so it's not very easy to use. One other agent which is getting a huge amount of press at the moment is remdesivir. This is a novel antiviral agent which was evaluated for Ebola disease and showed some evidence of effectiveness. It's currently used on a compassionate basis in the US. And there are some successful case reports in COVID-19 and ongoing clinical trials. This is an example of the compassionate use and the kind of cohort studies that we're getting here. The results on the right just show that 68% of the patients had some kind of clinical improvement while receiving remdesivir. But one thing just to point out is that it is funded by Gilead Sciences. And this medication is being heavily pushed by Gilead, particularly in the United States, but also elsewhere. And if you think this doesn't make a difference to the kind of quality of the research being conducted, just do a little search for Gilead's share price over the course of this outbreak to see the massive impact that this kind of press is having on Gilead's finances. All right, I'm going to have a brief look at tocilizumab, primarily the anti-cytokine agents. Theoretically, they dampen dysregulated inflammation, which might lead to cytokine storm. This is a very complicated bit of physiology. There's a really nice paper by McGonigal and colleagues in the Autoimmune Review looking at the mechanisms of how this works, and I've just included a diagram of this here. So it's basically a syndrome of hyperinflammation called secondary hemophagocytic lymphohistiocytosis, HLH, where the coronavirus stimulates macrophage activation, and through sustained levels of interleukins, particularly IL-1 and IL-6, ends up with an inflammatory process causes ARDS. They also postulate that there might be an element of excessive lung microthrombosis, which would explain some of the very high D-dimers and the fact that raised D-dimer is significantly correlated with mortality. So tocilizumab is an anti-IL-6 agent. It's approved for use in rheumatoid arthritis. It's currently been used in small case series in COVID-19 with some promising results, some of which are quite dramatic. However, none of these had comparator groups, so it's difficult to know what its true effect is, and studies are ongoing. Steroids. Theoretically, these could be used to dampen a host immune response as well. There's a lot of risks associated with corticosteroid use in various viral diseases. So there's a risk of adverse effects, including delayed viral clearance and the increased risk of secondary infection. This was relatively well studied for SARS and MERS, where it was found that there was no benefit from routine use of corticosteroids and an increased risk of complications. So there's also this nice systematic review and meta-analysis of 6,500 patients for influenza. And that study found an increased risk of mortality, 1.75, and an increased risk of secondary infections of nearly 2, which is very high. There are some case series from China looking at the use of this in coronavirus patients with ARDS. However, there's a lot of bias and residual confounding, so much more research needs to be done. But overall, caution with corticosteroids, and the advice is generally not to use it routinely. There's been some promising work looking at convalescent plasma and active immunoglobulins. This has previously been looked at for SARS and flu, and found that use of convalescent plasma in particular was associated with a massive reduction in overall mortality with relatively few harms. Although, again, they note that study quality was pretty poor, and lots of these trials had high risk of bias. There are small uncontrolled case series for coronavirus showing some potential benefit, and this is an area of exploration. Although identifying people who have had the disease and developed a good immunoglobulin response and then collecting their plasma is obviously quite a challenging task in this current environment, but it could be used for particularly very unwell people. I'm just going to mention supportive therapies very briefly. There are 2 very detailed sets of recommendations that have been published. The first one by Jason Poston and colleagues in JAMA, which has been organized by the Surviving Sepsis campaign and has a lot of interesting and important recommendations. And then secondly, this study by Jason Foy in the Lancet, which is part of the Asian Critical Care Clinical Trials Group. Both of these are worth a read, and there's lots of similar areas of crossover, and they go into a lot of detail about what does have evidence, what doesn't have evidence, and what is recommended in different scenarios. And it is very useful for guiding practice. Finally, I'm going to talk briefly about vaccines, which is our desperate hope to get us all out of lockdown at the moment. This is a nice diagram from a paper in Cell, which summarizes the development process. So on the left, we've got our coronavirus schematic. Here is the receptor binding domain of the spike protein that we saw attaches to the ACE2 receptor. These are the different vaccine targets that you can have. Ones that target the RNA, DNA vaccines, recombinant protein vaccines, which would look at these external surface protein, vectored vaccines, inactivated vaccines, where it's an inactivated coronavirus cell, or live attenuated vaccines. And on the right just shows the stages in development. So you've got to develop the molecule, go through clinical trials, get licensed, increase the production and distribution, work with a vaccine program administration system, and then develop immunity in the population. So it's a long and complicated process. But obviously the drive for this is very large, and so there's lots of potential vaccine platforms that are being looked at underway. So again, if you look down the left, these are the same technologies, as they call them here, but targets that they used before. So DNA inactivated, live attenuated, non-replicating, the protein subunit from the spike protein, replicating viral vector, RNA vaccine, or various other vaccine targets. And of note, at the time of writing, 2 of these were in phase 1 clinical trials. Non-replicating vector, which could be used as a single dose, which is good. It's reasonably fast to get production and potential for higher scale. And then the RNA vaccine that may require multiple doses is very quick to get to production, but it is more difficult to scale. However, this was a few weeks ago now, and as we've seen, evidence moves very rapidly. So there's this nice platform launched by the London School of Hygiene and Tropical Medicine, which is going to follow potential vaccine candidates as they move through the development pipeline. So this is worth keeping an eye on. And just to show that there's a large number in preclinical phases, particularly protein subunits, looking at spike protein and others on the COVID cells. They now note that there's 6 in phase 1 trials as far as they know, and 1 in a phase 2 trial, which is the non-replicating viral vector. So it'll be interesting to see how these develop over time. So that's it. That's my quick summary of virology, immunology, and therapeutics for COVID-19. Thanks for listening, and I will update this as new evidence emerges.